there have been several instances of archaic human admixture with modern humans through interbreeding of modern humans with Neanderthals, Denisovans, and/or possibly other archaic humans over the course of human history. Neanderthal-derived DNA accounts for an estimated 1–4% of the Eurasian genome, but it is significantly absent or uncommon in the genome of most Sub-Saharan African people. In Oceanian and Southeast Asian populations, there is a relative increase of Denisovan-derived DNA. An estimated 4–6% of the Melanesian genome is derived from Denisovans. Recent noncomparative DNA analyses as no specimens have been discovered suggest that African populations have a genetic contribution from a now extinct archaic African hominin population. Nevertheless, there still are some doubts about the recent admixture events among a number of researchers. Ancient subpopulation structure ancestral to modern humans, Neanderthals, Denisovans, and other possible archaic humans has been proposed as an alternative explanation for the observed genetic similarities. Neanderthals genetics equals. Through whole genome sequencing, a 2010 draft sequence of the Neanderthal genome revealed that Neanderthals shared more alleles with Eurasian populations than with Sub-Saharan African populations. According to the study, the observed excess of genetic similarity is best explained by recent gene flow from Neanderthals to modern humans after the migration out of Africa. The proportion of Neanderthal-derived ancestry was estimated to be 1–4% of the Eurasian genome. In 2013, the same team of researchers revised the proportion to an estimated 1.5–2.1%. They also found that the Neanderthal component in non-African modern humans was more related to the Mesmaiskia Neanderthal than to the Altai Neanderthal or the Vindiger Neanderthals. In the modern human population, at least those of East Asians and Europeans, the total introgressed Neanderthal DNA found spans about 20% of the Neanderthal genome. Although less parsimonious than recent gene flow, the observation may have been due to ancient population substructure in Africa, causing incomplete genetic homogenization within modern humans when Neanderthals diverged while early ancestors of Eurasians were still more closely related to Neanderthals than those of Africans to Neanderthals. On the basis of allele frequency spectrum, it was shown that the recent admixture model had the best fit to the results while the ancient population substructure model had no fit, demonstrating that the best model was a recent admixture event that was preceded by a bottleneck event among modern humans, thus confirming recent admixture is the most parsimonious and plausible explanation for the observed excess of genetic similarities between modern non-African humans and Neanderthals. A recent admixture event is likewise confirmed by data on the basis of linkage disequilibrium. Recent studies have shown a higher Neanderthal admixture in Eurasians. It indicated that most likely at least two independent events of gene flow must have taken place in two early modern humans and that the early ancestors of East Asians experienced more admixture than those of Europeans after the divergence of the two groups. This is estimated to have been an additional 20.2% of Neanderthal admixture in a second gene flow to East Asians. It is also possible, but less likely, that the difference was caused by dilution in Europeans by later migrations out of Africa. It may also be due to a lower negative selection in East Asians compared to Europeans. It has also been observed that there's a small but significant variation of Neanderthal admixture rates within European populations, but no significant variation within East Asian populations. A 2012 study found that North Africans have a Neanderthal admixture rate lying between that of Eurasians and Sub-Saharan Africans. It has also shown a great variation within North Africans themselves, depending primarily on the amount of Eurasian versus Sub-Saharan African ancestry. However, there are indications that their Neanderthal admixture is not solely contributed by Eurasian introgression. Neanderthal contribution has been very scarcely but significantly found in the Maasai, an East African people. After identifying African and non-African ancestry among the Maasai, it can be concluded that recent non-African modern human gene flow was the source of the contribution, as about an estimated 30% of the Maasai genome can be traced to non-Africans from about 100 generations ago. Through the extent of linkage disequilibrium, It was estimated that the last Neanderthal gene flow into early ancestors of Europeans occurred 47,000 to 65,000 years BP. In conjunction with archaeological and fossil evidence, the gene flow is thought likely to have occurred somewhere in western Eurasia, possibly the Middle East. No evidence of Neanderthal mitochondrial DNA has been found in modern humans. This would suggest that successful admixture with Neanderthals happened paternally rather than maternally on the side of Neanderthals. Possible hypotheses are that Neanderthal mtDNA had detrimental mutations that led to the extinction of carriers, that the hybrid offspring of Neanderthal mothers were raised in Neanderthal groups and became extinct with them, or that female Neanderthals and male sapiens did not produce fertile offspring. Recent studies found the presence of large genomic regions with strongly reduced Neanderthal contribution in modern humans due to negative selection, partly caused by hybrid male infertility. These large regions of low Neanderthal contribution were most pronounced on the X chromosome with five-fold lower Neanderthal ancestry compared to autosomes and contained relatively high numbers of genes specific to testes, meaning that modern humans have relatively few Neanderthal genes that are located on the X chromosome or expressed in the testes, consistent with the fact that male infertility is affected by a disproportionately large amount of genes on X chromosomes. It has also been shown that Neanderthal ancestry has been selected against in conserved biological pathways, such as RNA processing. Genes affecting keratin were found to have been introgressed from Neanderthals into modern humans, suggesting that these genes gave a morphological adaptation in skin and hair to modern humans to cope with non-African environments. This is likewise for several genes involved in medical relevant phenotypes, such as those affecting systemic lupus erythematosus, primary biliary cirrhosis, Crohn's disease, optic disc size, smoking behavior, interleukin-18 levels, and diabetes mellitus type 2. In a 2013 study, researchers found Neanderthal introgression of 18 genes several of which are related to UV light adaptation within the chromosome 3p21.31 region of East Asians. The introgressive haplotypes were positively selected in only East Asian populations, rising steadily from 45,000 years BP until a sudden increase of growth rate around 5,000 to 3,500 years BP. They occur at very high frequencies among East Asian populations in contrast to other Eurasian populations. The findings also suggests that this Neanderthal introgression occurred within the ancestral population shared by East Asians and Native Americans. Data from 2005 had previously shown that a group of alleles collectively known as haplogroup D of microcephalin, a critical regulatory gene for brain volume, originated from an archaic human population. The results show that haplogroup D introgressed 37,000 years ago into modern humans from an archaic human population that separated 1.1 million years ago, consistent with the period when Neanderthals and modern humans coexisted and diverged respectively. The high frequency of the D haplogroup suggests that it was positively selected for in modern humans. The distribution of the D allele of microcephalin is high outside Africa but low in Sub-Saharan Africa, which further suggest that the admixture event happened in archaic Eurasian populations. This distribution difference between Africa and Eurasia suggests that the D allele originated from Neanderthals. However, a 2010 study found that a Neanderthal individual from the Messina rock shelter was homozygous for an ancestral allele of microcephalin, thus providing no support to the theory that Neanderthals contributed the D allele to modern humans but does not exclude a possibility of a Neanderthal origin of the D allele. The 2010 Neanderthal genome study also could not confirm a Neanderthal origin of haplogroup D of the microcephalin gene. A 2011 study found that HLA-A02, A26/66, B07, B51, C0702, and C1602 of the immune system were contributed from Neanderthals to modern humans. After migrating out of Africa, modern humans encountered and interbred with archaic humans, which was advantageous for modern humans in rapidly restoring HLA diversity and acquiring new HLA variants that are better adapted to local pathogens. Morphology. According to a 1999 study, the early Upper Paleolithic burial remains of a modern human child from Abrigo do Lago featured traits indicating Neanderthal admixtures with modern humans dispersing into Iberia. Considering the dating of the burial remains and the persistence of Neanderthal traits long after the transitional period from a Neanderthal to a modern human population in Iberia, it was suggested that the child may have been a descendant of an already heavily admixed population. In a 2006 study, researchers found that the early Upper Paleolithic modern human remains from Pataramuairala of 35,000 years BP have the morphological pattern of European early modern humans, but possesses Archaic and/or Neanderthal features, suggesting European early modern humans admixture with rather than a full replacement of Neanderthals. These features include a large interorbital breadth, a relatively flat superciliary arches, a prominent occipital bun, an asymmetrical and shallow mandibular knot shape, a high mandibular coronoid processes, the relative perpendicular mandibular condyle to notch crest position, and a narrow scapular glenoid fossa. In a 2003 study, researchers found that the early modern human Nos1 mandible from P unregistered trademark Tyrannosaurus of 34,000 to 36,014 years BP presented a mosaic of modern, archaic, and possible Neanderthal features. It displayed a lingual bridging of the mandibular foramen, not present in earlier humans except Neanderthals of the late Middle and late Pleistocene, thus suggesting affinity with Neanderthals. Concluding from the Os1 mandible, there was apparently a significant craniofacial change of early modern humans of those from at least Europe, possibly due to some degree of admixture with Neanderthals. A 2007 study found that the earliest European modern humans and the subsequent Gravettians, falling anatomically largely in line with the earliest African modern humans, also had traits that are distinctively Neanderthal, suggesting that a solely Middle Paleolithic modern human ancestry was unlikely for European early modern humans. In March 2013, new data from the late Neanderthal jaw from the Messina rock shelter indicated possible interbreeding in late Italian Neanderthals. The jaw falls within the morphological range of modern humans, but also displayed strong similarities with some of the other Neanderthal specimens, indicating a change in late Neanderthal morphology due to possibly interbreeding with modern humans. A 2015 study reported about the Manot 1, a partial calvaria of a modern human that was recently discovered at the Manot Cave and dated to 54.7 A plus or minus 5.5 kbp. The finding represent the first fossil evidence from the period when modern humans successfully migrated out of Africa and colonized Eurasia. It also provides the first fossil evidence that modern humans inhabited the southern Levant during the Middle to Upper Paleolithic interface, contemporaneously with the Neanderthals and close to the probable interbreeding event. The morphological features suggest that the Manot population may be closely related or given rise to the first modern humans who later successfully colonized Europe to establish early Upper Paleolithic populations. History The hypothesis, variously under the names of interbreeding, hybridization, admixture or hybrid origin theory, has been discussed ever since the discovery of Neanderthal remains in the 19th century. Though earlier writers believed that Neanderthals were a direct ancestor of modern humans. Thomas Huxley suggested that many Europeans bore traces of Neanderthal ancestry, but associated Neanderthal characteristics with primitivism, writing that since they belong to a stage in the development of the human species, antecedent to the differentiation of any of the existing races, we may expect to find them in the lowest of these races, all over the world, and in the early stages of all races. Hans Peder Stensby in the 1907 article Races Studier i Danmark rejected that Neanderthals were ape-like or inferior, and, while emphasizing that all modern humans are of mixed origins, suggested interbreeding as the best available explanation of a significant number of observations which by then were available. In the early 20th century, Carlton Kuhn argued that the Caucasoid race is of dual origin consisting of Upper Paleolithic types and Mediterranean types. He repeated his theory in his 1962 book The Origin of Races. Stan Gutsch in Personality and Evolution and the Neanderthal Question develops a theory of Neanderthal Cro-Magnon hybridization, based not on an examination of anatomy but of his understanding of modern human psychology and society, which he claimed owes a significant debt to Neanderthal culture. Gooch's theories were dismissed by academia. Gooch refined his theory in Cities of Dreams and the Neanderthal Legacy. Denisovans, a 2010 study has shown that Melanesians share relatively more alleles with Denisovans when compared to other of the studied Eurasians and Africans. It estimated that 4% to 6% of the genome in Melanesians derives from Denisovans, while no other Eurasians or Africans displayed contributions of the Denisovan genes. It has been observed that Denisovans contributed genes to Melanesians but not to East Asians, indicating that there was interaction between the early ancestors of Melanesians with Denisovans but that this interaction did not take place in the regions near southern Siberia, where as of yet the only Denisovan remains have been found. In addition, a 2011 study has also shown a relative increased allele sharing between Denisovans and Aboriginal Australians, compared to other Eurasians and African populations, consistent with relative high admixture between early ancestors of Melanesians and Denisovans. In 2011, a study produced evidence that the highest presence of Denisovan admixture is in Oceanian populations, followed by many Southeast Asian populations, and none in East Asian populations. There is significant Denisovan genetic material in Eastern Southeast Asian and Oceanian populations, but not in certain Western and continental Southeast Asian populations, indicating that the Denisovan admixture event happened in Southeast Asia itself rather than mainland Eurasia. The observation of high Denisovan admixture in Oceania and the lack thereof in mainland Asia suggests that early modern humans and Denisovans had interbred east of the Wallace Line that divides Southeast Asia. However, in contrast to the previous results, more recent research found indications that mainland Asian and Native American populations had 0.2% Denisovan contribution, albeit 25-fold lower than Oceanian populations. The manner of gene flow to these populations is currently unknown. After Oceanians, it has been observed that particularly Southeast Asians in general have affinity to Denisovans. Findings indicate that the Denisovan gene flow event happened to the common ancestors of Aboriginal Filipinos, Aboriginal Australians, and New Guineans. New Guineans and Australians have similar rates of Denisovan admixture, indicating that interbreeding took place prior to their common ancestors' entry into Sahul, at least 44,000 years ago. It has also been observed that the fraction of near-Oceanian ancestry in Southeast Asians is proportional to the Denisovan admixture, except in the Philippines where there is a higher proportional Denisovan admixture to near-Oceanian ancestry. Reich al. Suggested a possible model of an early eastward migration wave of modern humans, some who were Philippine New Guinean Australian common ancestors that interbred with Denisovans, respectively followed by divergence of the Philippine early ancestors, interbreeding between the New Guinean and Australian early ancestors with a part of the same early migration population that did not experience Denisovan gene flow, and interbreeding between the Philippine early ancestors with a part of the population from a much later eastward migration wave. It has been shown that Eurasians have some but significant lesser Archaic-derived genetic material that overlaps with Denisovans, stemming from the fact that Denisovans are related to Neanderthals Euro, who contributed to the Eurasian gene pool Euro rather than from interbreeding of Denisovans with the early ancestors of those Eurasians. The skeletal remains of an early modern human from the Tianuan cave of 40,000 years BP showed a Neanderthal contribution within the range of today's Eurasian modern humans, but it had no discernible Denisovan contribution. It is ancestral to many Asian and Native American populations, but post-dated the divergence between Asians and Europeans. The lack of a Denisovan component in the Tianuan individual suggests that the genetic contribution had been always scarce in the mainland. A 2011 study, exploring the immune system's HLA alleles, suggested that HLA-B73 introgressed from Denisovans into modern humans in Western Asia due to the distribution pattern and divergence of HLA-B73 from other HLA alleles. In modern humans, HLA-B73 is concentrated in Western Asia, but it is rare or absent elsewhere. Even though HLA-B73 is not present in the sequenced Denisovan genome, the study noted that it was associated to the Denisovan-derived HLA-C1505 from the linkage disequilibrium. consistent with the estimated 98% of those modern humans who carried B73 also carried C1505. The Denisovans 2HLA-A and 2HLA-C allotypes correspond to common alleles in modern humans, whereas one of the Denisovans HLA-B allotype corresponds to a rare recombinant allele and the other is absent in modern humans. It is thought that these must have been contributed from Denisovans to modern humans, because it is unlikely to have been preserved independently in both for so long due to HLA alleles' high mutation rate. A 2014 study found that a EPAS1 gene variant was introduced from Denisovans to modern humans. The ancestral variant upregulates hemoglobin levels to compensate for low oxygen levels such as at high altitude ESA, but this also has the maladaptation of increasing blood viscosity. The Denisovan-derived variant on the other hand limits this increase of hemoglobin levels, thus resulting in a better altitude adaptation. The Denisovan-derived EPAS1 gene variant is common in Tibetans and was positively selected in their ancestors after they colonized the Tibetan Plateau. Archaic African hominins. Rapid decay of fossils in African environments has made it currently unfeasible to compare modern human admixture with reference samples of archaic African hominins. In 2011, after finding 3 candidate regions with introgression by searching for unusual patterns of variation indicating a different origin in 61 non-coding regions from 2 hunter-gatherers and 1 West African agricultural group, researchers concluded that roughly 2% of the genetic material found in some sub-Saharan African populations was inserted into the human genome approximately 35,000 years ago from archaic hominins that broke away from the modern human lineage around 700,000 years ago. After a survey for the introgressive haplotypes across sub-Saharan populations, it was suggested that the admixture event happened with archaic hominins that possibly once inhabited Central Africa. In 2012, researchers studied high-coverage whole genome sequences of 15 sub-Saharan hunter-gatherer males from 3 groups: 5 pygmies from Cameroon, 5 Hadza from Tanzania, and 5 Sandal from Tanzania, finding signs that the ancestors of the hunter-gatherers interbred with one or more archaic human populations, probably over 40,000 years ago. They also found that the median time of the most recent common ancestor of the 15 test subjects with the putative introgressive haplotypes was 1.2 1.3 MIR. Alternative hypotheses. In a 2013 study, the researchers suggested that the observed genetic affinities between archaic and modern human populations are mostly explained by common ancestral polymorphisms and not admixture followed by genetic drift, explaining that the differences in the observed genetic affinities among modern human populations are most likely a result from different retention rates of these polymorphisms among the modern human populations. However, they also stated that the study did not rule out archaic introgression to modern humans. See also, multi-regional origin of modern humans, Neanderthal extinction, recent African origin of modern humans. References